Welcome to Health Facts with Chinwe, and welcome to our second episode in the vaccine series. The first episode we talked about vaccines, what vaccines are and how they work, and especially how they work with your immune system to protect you from infectious diseases. I'm going to post the link in the comment section and for you to refresh your memory, and if you're just joining us, for you to catch up on this very informative topic. Remember that vaccines work by triggering your antibodies that would help protect you from falling sick. In this video, I will be talking to you about the different types of vaccines and how they work, and also the different measures that are put in place to ensure that your vaccines are safe for use, and a few success stories of vaccines. So you may wish to subscribe to this channel if you haven't done so, and also press the notification button so that you're always notified of our weekly videos. So recall in our previous video that we said that vaccines are made from the pathogen. So by the pathogen here I mean the disease-causing organism, either the virus or the bacteria. So just remember that pathogens are the virus or the bacteria that causes disease. I'll be using pathogens subsequently, so just try to grab it. Now, the vaccines are made from versions of these pathogens, and they are made such that the dose that is being used is enough to cause your immune system to produce antibodies, to trigger your immune system to produce antibodies against this, either the virus or the bacteria, such that when your body gets exposed to the main thing in the future, your body is able to recognize it and your body is able to destroy this pathogen without you falling sick. So that's a bit of a background of how vaccines work. And so these different versions actually represent the different types of vaccines. And there are about 6 types of vaccines, and I'll explain each of these types. The first type of vaccines is called the live attenuated vaccines. So these type of vaccines are derived from the pathogens, but in this case, the pathogens are weakened under laboratory conditions. So when they are administered to you, they are able to multiply, but since they are in their weakened form, they are not able to cause the disease, but they are able to trigger your immune system to produce antibodies. These type of vaccines are one of the oldest vaccines, and they've been available since the 1950s, and they've been used successfully. However, in some rare cases, these kind of vaccines have actually caused disease, but it's mostly amongst people who have very low immunity, people whose immune system is compromised, or persons who are under immune suppressing drugs. But the main purpose of a vaccine is not to cause disease, as some persons will say. The main purpose of a vaccine is to train your immune system. And so the examples of vaccines in this category are oral polio vaccine, which is being phased out now, and we have the BCG vaccine, which is used against tuberculosis. We have the rotavirus vaccine, which is used against diarrheal diseases in children, and yellow fever vaccine. So the second type of vaccines that we have available is called the inactivated vaccine. And so unlike the live attenuated, this one is a killed version of the pathogen. So it is killed, completely killed, and then used to make the vaccine. So our influenza vaccines or the vaccines used against pertussis, the latest version of the polio vaccine. So another type of vaccine is called the subunits. So this is a particle that is being taken from the pathogen and it is used to make the vaccine. So this particle is usually the part of the pathogen that is responsible for causing disease, either the part that enables the pathogen to enter into your cell or to cause you to fall sick. So that part that is responsible for causing disease is usually used to make vaccines. So the vaccines in this category are the hepatitis B vaccines, meningitis vaccines, and vaccines used against pneumonia. So vaccines can also be made from the toxic substances that are being produced by the pathogens or toxins. And these type of vaccines are called toxoids. So the toxins are usually firstly rendered harmless in the laboratory and then used to make the vaccines. And the common toxoid is your tetanus toxoid vaccine or your diphtheria vaccine. More recently, in the past few decades, we have seen newer versions of vaccines become available. Example is the mRNA vaccine, which is one of the COVID-19 vaccines that has become available, and another type is the viral vectors vaccines. So like we explained that a vaccine can be made from a part of the pathogen that is responsible for causing the disease. The mRNA vaccine, or for coronavirus for example, the virus has on its surface spike proteins, and these spike proteins are responsible for causing disease in humans, and these spike proteins enable the virus to penetrate into our cells and cause disease. And so the target here is the spike proteins. And so unlike the older vaccines where they would have made a version of the spike proteins in the laboratory, or they would have made a vaccine from the spike proteins, for the mRNA technology, it uses a genetic material or a genetic template. This genetic material is used to instruct your cells to produce the spike proteins. So these spike proteins are actually produced in the body. This genetic material contains a template for the spike proteins. And so when the vaccine is being administered, this material gives an instruction for the body to produce those spike proteins based on the template. And obviously this template was made from the spike proteins or from the structure of the spike proteins. And so when your body produces these spike proteins, your body is also able to identify that these spike proteins are foreign, and then your body develops antibodies against these spike proteins. Your body destroys the spike proteins, and then that also means that when you eventually become exposed to the main virus, the coronavirus itself, obviously which contains spike proteins, your body is able to destroy the coronavirus. You should also note that once this vaccine has done its job of instructing your cells, the vaccine, just like any other medicine, is also destroyed and it leaves your body immediately. And so the spike proteins are being destroyed, and the vaccines or the material that is contained in these vaccines are also being destroyed. And so you just have your antibodies circulating and waiting for future exposure. You must also note that these vaccines, this type of vaccines, even though they contain a genetic material or they contain genetic materials, they do not alter your genes. They do not change your DNA. It just comes in to instruct your cells. And this technology actually has been in existence for over a decade now. Like, scientists have been conducting several research on this particular type of technology, or they discovered that this type of technology actually is less expensive and it is easier to produce than actually making the spike proteins in the laboratories, purifying it, and going through all those rigorous processes. So this is a much more easier, it was less expensive, and also is able to trigger a much more stronger immune response compared to the traditional vaccines. So the last type of vaccines, which is one of the newest versions, is a viral vector vaccine. It's also similar to the mRNA vaccine, but in this case, a harmless virus is used to smuggle in this genetic instruction into our cells. So basically the virus carries these templates to produce the spike proteins, and it is delivered into the cells and obviously helps to trigger the immune response. The commonest virus that is used is the adenovirus. Adenovirus is responsible for causing the common colds, but a lot of people, almost everyone, has immunity against the adenovirus. And again, the variant that is being used is a very harmless one, and it just helps to transport this genetic instruction into the cells. Apart from the COVID-19 vaccine, the viral vector technology has also been used in making vaccines against Zika virus, against HIV, although that's still undergoing clinical trials, and against virus that causes Ebola disease. I just hope that you are not overwhelmed with all of this information, and if you are, please bear with me. I'm just trying my best to give you all the information, the right information, so that you are well informed. So before these vaccines are being approved for use, they actually undergo rigorous processes, rigorous studies, and these studies are called clinical trials. They undergo clinical trials before they are approved for use. So clinical trials follow a rigorous process to ensure that these vaccines are not just effective against the disease, but they are also safe. So safety is one of the priorities in every clinical trial. A typical clinical trial has 3 major stages. The first phase of a trial is where what we call the candidate vaccine, so that's the vaccine that you want to test, is being tried on animals. But before it's being regarded as a candidate vaccine, it is first of all tested in the laboratory. So it is being tested to see if it has some effect, against the virus. And when it shows some effect, it now moves to the stage where it is tested in animals. And so this is the first stage. The first stage where it is tested in animals, firstly, to check for, first of all, the safety in these animals. When it passes this stage and the safest dose is determined, it's being tested in a very small group of humans. So it moves to the next stage where it is tested in humans. And the aim is also to check if this tiny dose that was identified in animals is also safe in humans. And then the third phase is a phase where that safe dose that has been identified in the previous stages is now being tested in a much more larger group of people. In fact, this would run into thousands of people, and most times it is conducted in multiple places, so in multiple countries. So these will be happening at the same time among different kinds of people, different colors, different age groups. And still at this point, the safety of this vaccine is also being monitored, as well as efficacy. When this phase is completed, then the findings, the data, are now being submitted to an independent review group. So this independent review goes through the data that was gotten from the phases of the trials, and at this point, some vaccines, the vaccine can be approved or not approved. So depending on whether they find this data suitable enough to allow the vaccine gain approval. And the vaccines actually even fail at different points. Like not all vaccines that start up from that first phase usually get approved. Some of them fail at the first phase, some fail at the second, and even at the third phase, some of these vaccines fail. So when the vaccines gain approval, they can now be used for the general public. And even when the vaccines have been approved for use in the general public, it is also being continuously monitored for both safety and efficacy. So the monitoring never really ends. It continues even while it's being used now in real life scenarios. So you can see the efforts that is being put in place to ensure that these vaccines are not just effective, but they are safe for use. So in summary, vaccines work by training your immune system to produce antibodies, and these antibodies are your body's defense system. So they produce antibodies against specific pathogens to protect you from the diseases that are caused by these pathogens. And these vaccines usually go through rigorous processes to ensure that they are safe and to ensure that they do not cause harm. And you must also note that vaccines actually are produced for the sole purpose of preventing infectious diseases, and they are not made to reduce the world's population. They are not made to harm you. They would not cause infertility, and they have been known to prevent infectious diseases, and they have been known to prevent infectious. A lot of lives have been saved due to vaccines, and they continue to save lives. So I hope you learned something today, and if you did, as usual, please leave me a thumbs up, and also don't forget to subscribe if you haven't done so, and also kindly share this video with people around you. So till I see you next time, as usual, stay safe, keep well, and always stick to scientific evidence. Thank you so much for watching. Bye bye.